Hello everyone and welcome back to our drug discovery and In-Silico tools series. Today we are entering to the detective phase of our drug design, as we have previously discussed about the introduction to the web servers and the tools that we can use in our drug discovery and designing and the projects and also the steps involved in the drug discovery and designing, the target identification, validation, then lead optimization, and then the interaction through docking process, then visualization, then further clinical and preclinical trials and clinical trials. And our today's lecture is about target identification and the validation using bioinformatic tools. Before we design a drug, we need to know who we are fighting against. You can't win a war if you don't know your enemy, right? So, target identification means finding the specific molecule inside the body that's responsible for a disease. Think of it like this: if the body is a city and disease is a crime, we are trying to identify the criminal behind it. Sometimes it's a protein that gone rough, sometimes a gene that's misbehaving, and our job is to find that bad guy. So identify the culprit, define the target, the risky bet. Choosing the wrong target means you are developing the wrong drug. A high stakes failure is time and capital. So, first we have to identify the culprit. Pinpointing the molecular agent, the protein, enzyme, or a gene, or any external pathogen if it is. Then we have to also if we are targeting directly the pathogen, then we have to select a protein or a gene inside the pathogen. If we are targeting the protein and gene that enhancing that pathogen or a disease which is occurred without any pathogen, and because of the misbehavior of our protein, enzyme, or any kind of gene, then we have to select the protein or gene within our body, like human body. So identify the culprit, then defining the target. The target is any molecule whose function can be modulated by a drug to achieve a therapeutic effect. So the risky bet, I have explained it, that a high stakes failure is time and capital. So it's a risky if you have selected blindly a target and it's not a specific target. We have developed a drug and it's not effective because we have selected a wrong target. Our first step was wrong. We stepped into the drug discovery in a with a wrong step on our first step. So moving forward, that why this initial step defines success, why it's matter. Imagine you are trying to sweat a mosquito in a dark room, and that's drug discovery without target identification. You'll waste energy, time, and maybe break a lamp. So but once you identify the target, you can focus your entire effort precisely on that molecule. That's what makes modern medicine so efficient. So, the initial step defines success in drug discovery. A promising drug is intrinsically linked to a high quality validated target. If the target isn't crucial to the disease mechanism, the resulting drug will fail, regardless of how potent it is in a petri dish. If it is not potent in our in vivo working, in preclinical or clinical trials, then it's waste. This step saves millions and accelerates the path to clinical trials. Then we have wasted the millions or even the billions of dollars in our whole drug discovery. If we have done it on the computer and also done it in the in vitro, like I have mentioned that how potent it is in petri dish. If it's potent in a petri dish even, but it's not potent to the in vitro clinical trials, then the whole process will be a loss. millions or billions of loss. So minimizing waste, enhancing precision, strategic direction provides a clear molecular mechanism for drug design process. Imagine shooting an arrow without knowing where the target is. Then you will make someone else blind if you do blindly target identification. So moving forward, the usual suspects, types of targets, we have common classes of drug targets. Who are our usual suspects? Enzymes that catalyze wrong reactions and receptor that overreact. You're overreacting, and that's why and channel the leak signals. Even in our take an example of a relationship. If someone is in a situation and the front real is our receptor who have to accept our behaviors, and if we react minimal to anything and they overreact to it, then it's harmful for our relationship. That's why the receptors that overreact, so these would be the target. Not every enzyme and every receptor we can select it. Oh, this receptor we selected. No, we have firstly to identify that which receptor that this disease is causing because of this, and this enzyme is receptor, and this in this way this goes through. So we have to find the specific one enzyme and receptors. So the ion channels that leak signals, and sometimes DNA itself. Each of these targets have a unique role, and our future drugs will need to calm them down, block them, or sometimes destroy them entirely. Discuss the slides in a short way because we have to go through the web servers and to identify our target and to validate it. And the digital lab assistance is the NCBI, where we'll get the gene, and where we'll get the protein and all its genes information. And on the UniProt we'll get the whole protein structure and whole protein sequence, and also the amino acids and its functionality on the UniProt. And on string we'll see the interactions of the protein and of the gene with the other, and it's also the working of that protein. It will be viewed on the string search tool for the retrieval of interacting genes, proteins, and this one. So the main tools in for our this lecture will be NCBI, UniProt, and the string. So the step-by-step target identification workflow: first we have to search for the gene on NCBI, then analyze protein on UniProt, then map of interactions on string, and then select the target. um, Prioritize disease, variant, protein, candidate. So that's the pathway that we'll follow in our journey towards the web servers. So this is the pathway that we'll follow towards the web servers while searching a gene, and then the protein on the UniProt, and also the map interaction. So that's also the initial search. Any disease will capture it, and then we'll go through this. Any gene, then the protein that will be linked to it, and then we'll functional deep dive on the UniProt, then the contextual mapping on the string, the protein's neighborhood, identify binding partners and confirm its role in specific pathway, then the candidate selection based on the pathway enrichment, and that's why. So critical checkpoints is target validation. Now validation means we confirm the protein is truly responsible for the disease.
not just hanging around. If it's conserved across species and involved in critical pathway, that's a big hint. Imagine trying to stop a heist. If this guy shows up in every camera footage, yep, he is the suspect. So that's the target validation. We have to confirm the causality, therapeutic linkage, and validation tools to the critical experimental proof, and also reach to the gene locations, and in this way will validate the target. So case study, we have to study, we'll take a disease says Alzheimer and start the hunt. We'll go from gene protein interaction, and by the end we'll have our confirmed target molecule that play a key role in the disease. So we move towards our web servers. Here you see we first go to the NCBI site. It will take time to load it, and we have to locate the gene here as our case is Alzheimer. So paste it here, Alzheimer. I've copied from there the spelling mistake. So search for it, and also open the UniProt simultaneously. It also take time. So here you go towards the genes. 3938 genes it's showing. So we go towards the gene because we have to um, locate the gene information and also the protein. So our protein here we have to locate the gene is APP. So first we set the relevance, load it, yes, name, by name, then it will be easy to locate the gene by name. I was on the next page, ABC, it will go in this way. So the next, loading, we jump to the page 5. We will start the alphabetically the AP. It's still AD, we have to go to the AP. So page 10, it's AP, but it's APP, we have to locate APP. It's still APO, it's APO, we have to locate APP. Here it comes, APP, amyloid beta precursor protein. So we select the first one, Homo sapien, humans. Chromosome 21, and lyses are AAA, and we see the gene information here. Yes, it's open. You can download datasets here. The official symbol is APP, the amyloid beta precursor protein, and primary source is also mentioned here. You can also, the blue one is a link, you can click them and see them. It's a protein coding gene type, organism is Homo sapien. So if we go towards the UniProt, we have the protein coding gene APP. From the Homo sapien we write it human. It's loading on the UniProt, and Here the table or cards, we select the tables. View results. Yes, we have to select the amyloid beta precursor protein, as this one is the amyloid beta precursor like protein and amyloid beta precursor protein A4AD1, as we have seen there. AD1, A4, and then we go through this amyloid beta precursor protein. I was late in the lecture, so here it is the whole protein structure. Now here's our protein complete with sequence, structure and function. It's like checking someone's LinkedIn profile. You can see their entire career and mistakes in one place. You can see the annotation, amino acid chains, organism, functions, and download also in the sequence of amino acids in the FASTA format. We will load and we go through the profile of our protein. Here is its whole function mentioned, and the gene ontology is also mentioned. You can see the whole gene structure, that which portion works for which thing. Keywords, enzymes and pathway database, names and taxonomy, organisms name, subcellular, C83, miscellaneous, disease and variants. Then the number first disease involvement is Alzheimer disease. So that's a whole profile where you can get the whole information about your selected protein. So you are viewing the expression in time. FASTA format. You can also select it and copy it and do the blast or anything. We'll do in our next videos. In our lecture I'll show you that how you can do the blast. So moving toward our third web server, there's the string for the interactions of protein we have selected. So it's opening string. So first hit the search. Here we have to write the protein name APP, capital P, and then the organism name. Open up. Let's take time. This the Homo sapien. It's still not selecting. If it can't select, then we'll write it manually. It's can't selecting. Again click it. Can't selecting. So we write it manually. Homo and the sapiens. Oh, that's we are. No one else. We have to hit the enter. It's searching. Here we just have to select the protein and then hit continue. We don't have to do anything. We have selected the first Homo sapien. It is the protein. Look at this colorful web. This is the social network of APP. As you have the number of friends on your Facebook and LinkedIn connections, it interacts with beta secretase and parsinilin, non troublemakers that cause neurodegeneration. So this confirms APP is a legit disease target. You can go through all these colorful they have given, and also the protein structure you can go through. As you have seen, the red one lines are moving the gene structure, and they are giving us the sequence wise structure, the end of the gene. Here it is. So that's the protein structure, and here's the explanation that to understand the colorful web, and also your input was and prefer functional partners, homo sapien taxonomy id, and here the nodes, color nodes, query protein, and white nodes, second shell interaction, empty nodes, and network node represents. So these were the legends. And then cluster, if you want to show the clusters, exports, and analysis that are made, number of nodes it has 11, number of edges 33, and go through them. So this is to we have validated it that that's the culprit, that's our criminal that we were searching for. So biological processes involved, you can see the whole interactions basics here on the string. So viewers, you can select the which thing you want to view, experiment, network, databases, or anything. So that's the main legend we have to. So that's the. So moving back to the lecture, the real-time applications into the databases, as we have viewed in our previous video on the web server's journey, you have seen it. So the true power of bioinformatics lies not just in assessing data, but in efficiently connecting these three sources to build a routine. So these were the our future is informed discovery. Identify the culprit. It's about finding the single molecule. We have discussed it before, digital assistance, and so the next lecture for this series will be molecular docking basics. We'll also go through the hit identification, because we have identified or found the lock, but now we have to identify the key, and also we'll discuss the molecular docking basics. Then we go through the servers and the softwares that will be used for molecular docking, lecture by lecture. In every video we'll explain every web server and the softwares that are used for the molecular docking separately in every video. So don't forget to like, comment your questions, I'll guide you, and also don't forget to share it with your fellows and with your research fellows and with your well-knowns who are doing their research on computational uh, drug discovery and designing the projects. So be with us, like us, and follow us on the Facebook, it's Hammad Bashir Khan, and subscribe us on YouTube, it's Bioinformatics by Hammad. You can get the new updates there, and if you uh, follow us, we'll give you. Also, you can book a lecture for a group or individually. There you can get the training uh, for the whole in a single lecture for target validation to the clinical trials. And be with us. Thanks for your time. Allah Hafiz.